to start off with, I want to say we have this pipeline here to understand what's going on a little bit under the hood, such that we can better understand what are the pieces that we now fiddle together when using the BoltzGen repository that's on GitHub, and how to control them and what's actually happening. And at the very beginning of this pipeline, there's this design model, this generation model that we use for designing binders. The main way that we would use BoltzGen is that we want to design some binders. So I guess the thing here at the bottom left. And now I will first talk a little bit about how this foundation model works for designing such binders, for generating them, and then a little bit about how we can control it and control its generation process to then end up with designs that actually are at the right binding site and fulfill all the constraints that we might want them to satisfy. And with that, I have now this main task laid out of we have some target description given of some target protein. It could also be a target small molecule. And then we want to generate this binder here in yellow, and we will be sticking with this color scheme to bind to it. And now what we do in this generative model, right, you probably know RF diffusion and the likes. We start from complete noise, but now for both the binder and the target, and then iteratively update this with this type of generative model, the fusion models, such that we in the end end up with this 3D point cloud that encodes our binder design. And then we also do this for the target. I'm just not visualizing it here for the target. So the model can also reason a bit about how the target is changing its shape under binding the binder that we're simultaneously designing. The only other piece that I'm going to say about this core generative model is how it is, or what is the data it is trained on. And we take everything that's in PDB. We also take some distillation data, some additional data, and then ask the model to randomly generate these yellow regions that are randomly sub-selected from the overall complex. And right, if we have a dimer, then maybe this is now a binder, a protein that binds to another protein. And maybe if we have some protein RNA complex, protein DNA complex, we end up tasking the model, training the model, supervising it to design DNA, protein matter that binds to DNA. And also in our monomers, we have this algorithm that sometimes selects a sub-region of the whole protein to be deemed as the binder, while the rest is being considered as the target, and we task it to design this. Sometimes during training, you also would have a uniformly at random selected set of residues in the protein that is considered as the binder or as the design. This then means that at inference time, you can also use the model for things like we have an interface of two proteins, and now I just want to redesign 3-5 residues in there and then task the model to do such a thing. But with that, let us get to these control options. And I'll now just step through what we can do a little bit. What are these control knobs? And then I will show how can we dial these control knobs and combine them to then end up doing many different tasks. And now the thing that I'm highlighting here is that we can take an existing protein like some nanobody, for example, and take its scaffold and redesign the CDR regions to bind to a certain target. Then we can also specify that there should be a covalent bond within a peptide, for example. For example, also a disulfide bond if we want the peptide to be more stable, or a head-to-tail cyclization. And this then might be what we want for your more stable peptide that has less entropic penalty when binding the target that you're interested in. And of course, you can specify the binding site, but something else that's maybe not seen so often, you can also say that this is a particular residue that I do not want to interact with. So, for example, in a collaboration with Carol and Bertozzi, we're using this to say we want to be selective to this glycan, and we only want the design to interact with the glycan and not to interact with the rest of the protein, such that we only bind this whole target if it actually has the glycan attached to it. And lastly, we can, of course, specify what is the structure of the target. And there are also only partial regions of the target, if you're interested in that. And another thing you can actually do is you can say this region, I want the structure to be specified. This region, I want the structure to be specified. But with respect to each other, the structure should be free to move, things like that. And now we, for example, use this then to we're designing peptide binders against intrinsically disordered regions, where we leave it up to the model to decide how the intrinsically disordered region will fold up upon binding the binder that we simultaneously design.
Okay. Just out of curiosity, how do you get this controllability? Do you have to do this during training? So do you train for a specific case?
For us, all these control options that I talked about now, they are just fed in as covariant inputs to the model. So during training, the model sees all these cases. right? And then we have our training data here. And let's say we're in the case on the very right, where we are now tasking the model to see all the blue region as the target and the yellow region as the binder that we're designing. And then there's also an algorithm that will randomly pick sub-regions of the blue part to have the structure specified and then feed the structure into the model. And this is fed in as pairwise distances, but only of the backbone and the side chains, they always remain free to move. And also,
you only have one final model, right? Because RF diffusion, for example, has several models that were a little bit tuned for different tests. But do you have just one set of weights here?
Yep. It's one model that's trained in all these different scenarios. And we do so deliberately. And we do not train like an antibody model and peptide model and so on. We try to train across all modalities because we want our model to generalize, right? And to generalize, you need to reason about patterns that transfer, like geometry and physical chemical interactions. And these patterns, they appear in all modalities. And thus, training on all these modalities isn't just nice because you end up with one model that can do all the modalities, but also because it further aids us toward generalization if we get our model to reason about such generalizable patterns. And with this, I will get to now the end of laying out this sort of first steps of the generative model and how this then would actually go in practice of using it if we execute the code. And there we then would run this diffusion model to generate, for example, 60,000 designs. And this is a significant time investment and compute investment. And for example, if I run this against a target with, say, 100 residues, then it'll take on 8 GPUs around 2 days to generate these 60,000 designs. And it is a significant compute investment for sure. Then we have this inverse folding step. I think most of us here know inverse folding models. And we just have now another inverse folding model in the BoltzGen pipeline that can handle DNA, small molecules, and so on. And it's trained very similar to soluble MPNN, in case you know that. And then we have the standard refolding step to produce compute scores that we use to distinguish and to rank our designs, where we take a folding model, in our case Boltz-2, to predict the structure from the sequences only. And then from that predicted structure, we check, does it match the original design structure? And we save these IPTM scores and all these interaction metrics based on the confidences that come out of these folding models. Then, actually, for small molecules, if we design binders against small molecules, we have an affinity prediction step that I'm skipping here. And lastly, we compute all sorts of more physics-based metrics where we count, for example, the number of salt bridges, hydrogen bonds, the interaction surface area. This is similar to what's happening in Bindcraft, if you're familiar with that model. And we're computing many additional scores. And at the very end, we combine all these scores into a final ranking where we have some ranking mechanism running that essentially makes sure that your design needs to score high across all metrics and not just high at the sum of metrics. Okay, and then you have your final ranking and you test the top 10 in the wetlab, for example. You want to do. But just this, let us have a step through of how it would work in practice. What is the input to this pipeline where you say what you want to design and how that would look like. So the first step I would do if now say I want to design a binder against this target on the left and have a peptide that displaces the DNA that usually is in the target, such that this protein, which is telomerase that elongates this DNA, it can no longer elongate the DNA. And what I would do then is now, because there's a structure available, go to PDB and download the CIF file for it. Then I tell in my design specification language, I call this now, which are just YAML files, .yaml files, I go ahead and say, this is the file, the CIF file that contains the structure of my target. And then I say, I look into the structure of my target and I see, chain A corresponds to my target. And this is actually what I want to model as the target. So I tell in my design specification language to include from this complex, please, chain A as the target. And then I also want to select the binding site. So I go into my file and I look at, this residue here is index 341, the other is 346. So I say the binding site should be these residue indices. And I could also then specify not binding, for example, right? If I want to say that another region should not be interacting. Then we have this. And lastly, I go ahead and here in this scenario, now just specify the structure of all the targets. So the whole structure of the target is fed as input because here it's coming from a PDB structure. We know it well. We don't expect there to be much flexibility. And then I now come to the piece that we want to design here. And in this scenario, I say I want a peptide that has a random number between 3 and 7 residues on the left here. And then we have a cysteine. Then we have 6 other residues that are being designed by the model. And we have another cysteine. And then we have, again, a random number between 3 and 7 of designed residues. And then I want these to be covalently bound to a small molecule because I want to design this type of binder that is what's called a helicon or a stable peptide. And for that, I then say I want to, in this soup, also include the small molecule. So I add this as another entity here, this ligand that's called WHL. You could also specify this as a smile string. Here, we specify the CDID. And then we say we want to have a covalent bond between chain B, our peptide, and chain C, our small molecule, between the right atoms, right? And this is then our finished design specification file or so. And this is what we then put into this whole pipeline. It generates the 60,000 designs, ranks them, and so on and so forth. And we get out a final designs. Now,
can I ask you a quick question? So how do you know which residues are, if there's a varying number of residues on the left and the right, how do you specify the indices of the residues that are supposed to be connected?
Yep. What it always assumes is that the lowest number of residues is what you choose. So if you look at the, I'll just jump out here so I can use my mouse. You look at the 4 here, right? We're saying at chain B, position 4 is the cysteine we want to be attached to. So we assume always it's in specifying the indices. And of course, it will be correct in the model will know how much randomness or how many random residues there were sampled and then put it at the correct cysteine. But when you specify it, you just say the 4th one, those 3, 4, and then you say the 4th one. And then we have 3, 4, and we have another 6. So that's 10. And then this cysteine is the 11th, and we say 11. And now everything might be nice, right? We run the model, we get out our designs, and maybe they're binders. But what I do actually in practice is that I don't right away run these, generate these 60,000 designs. And maybe the slides now could be ordered in a better manner here, but I just want to right away get to this limitation because it is not a plug-and-play solution that you make your design specification and it just works. You usually have the specification, you run it, you get out your designs, and you run it on a small scale. Maybe you generate 10 designs, and you look at the structures. Do they make sense? Does it go to the right binding site? And then you see, maybe I have here my target, and I wanted to design a nanobody against it, and I wanted to interact with this binding site, and then the nanobody always goes here, but I have a very low pass rate with my final filters, and almost never the structure folds up into the structure that we designed when we do our refolding step. So I then see, it's trying to get in there, but it's not getting in there. So let me actually not specify the structure of this part and have this part be flexible to the model. And then maybe now 100% of designs fold up with RMSD less than 2.5, and we run the thing again and see what comes out, and it happens to work well. And then I go to the big case and run this for 60,000 designs, for example. Yeah, and now I just have another last example here, because I know many people are interested in nanobodies. And here is what it would look like if we, for example, had this PMHC complex on the left, where we have the peptide bound to the MHC. And then we say we want the target to be this MHC piece from the, if you look at number 3, this MHC piece from that file that contains the PMHC complex. And then we say the peptide is what we want our design to interact with, what we want our nanobody design in this case to interact with. And the nanobody is specified in this yellow region, where what we do is you can specify your frameworks or your scaffolds of nanobodies that you want to use. And we have also 4 defaults frameworks that are some FDA-approved nanobodies and some scaffolds that have good developability properties. And then what the model will do, it will randomly sample from all these 4 possibilities of scaffolds. And also the CDR3 length can be between length 4, let's take something more realistic, between length 8 and 15. You can choose what range of lengths you sample there if you have some prior knowledge, for example. Yeah.
And this length that you're sampling, is that specified in this YAML file, or do you specify that in the universal language?
So that is specified here. Sorry.
Go ahead.
Okay, I will go and say that it is specified in this, for example, 7EOW.yaml. And we can look at the things if we want to and make this a bit more interactive. Sorry. Yeah. And so this YAML file contains these lengths and says, for example, in my nanobody that I extract from this and that CIF file, because we have the structure of that nanobody, I want the region between these and that residues that correspond to the CDR region to be deleted and replaced with a region of length 3 to 5, 3 to 8 for the CDR1, if that's a realistic number. I don't really like it. Now we can, for example, see this here, 7EOW, where we see this, right? We, in this file that defines the nanobody scaffold, we say, we take the CIF file, we extract the chain that corresponds to the nanobody, and then we say, we design these regions, which correspond to the CDR3 regions, and say something like, for example, that the structure of the backbone of the scaffold stays fixed, and the structure of the CDR regions is flexible. We remove also the CDR regions, and we insert regions that are being designed of the appropriate types that we desire. Yeah, and that's about it that I wanted to present, and would love to move to any type of Q&A that we have after saying that, of course, this project was made with many collaborators, and the wetlab validation can be found in the paper.
Great. Thank you very much for this talk. And thank you also for joining from the airport. So that's not the easiest environment to present. So thanks again. Yes, please paste questions in the chat. And I'll start with maybe a general question. Do you think you would benefit by replacing the refolding step with, I don't know, AlphaFold 3 or something that is completely unrelated to the way you train? Or is that, do you think the models for the refolding and design are already orthogonal enough?
Yeah. Right, we also know that the AlphaFold IPTM is a bit better at distinguishing, for example, antibody binders from non-antibody binders on retrospective data where the designs do not come from machine learning models or from machine learning designs. In general, it could just be better because of that as well to use AlphaFold scoring instead of Boltz2. But we use Boltz2 here such that we have, because I expect the difference to be small and such that we have something that anyone can just run, and you don't have to worry about any IP stuff. You can do whatever you want with what comes out of this pattern.
Yeah, for sure. And then, since this is about workflows, how much time did you spend designing the YAML layout? Did this just evolve like organically?
Yeah, it's evolved organically a little bit by I have to run the model, and I have to run the model for 50 different collaborators, and I need it to be easy for me to run the model. And then this is how this changed a lot and over the time, and now this final specification language is somewhat stable, but it's also still changing, and we're adding new options all the time. Telly Portnoy right now in our lab, she is doing a lot of that, where she's having very creative projects of having complexes where she needs to, for example, do symmetric redesign, because we have two symmetric chains, and now I want them to change the conformation to always be bound together like this. And then we take the interface here and the interface here and say we want them to bind to each other. And now she added this symmetric design option there, for example.
And what would be the best place for documentation on this universal language? Is this on the latest version on GitHub, or do we read the source?
Yes, that's for sure on the GitHub here. And there we have a very high level step through this GitHub. And then we have a more detailed explanation in these examples. So here we have a whole bunch of examples of, you want to design, for example, a peptide that binds a disordered region, or you want to design a disulfide-bonded peptide. And then down here we have all the options documented and laid out.
Very cool. Now we have a question from the chat. And the question is, do you have any plan to add features for screening against off-target decoys? So let's say I want to design a binder, but make sure it doesn't bind 3 other proteins. How would you do that? Would you do this in a different run, or filter afterwards, or could you do it at the same time?
Yeah. So what we are doing there is, our reasoning is, if you now generate and filter post hoc, right? And now you do this filtering, not just for one condition, binding against A, but also for not binding against B and C and D. And then your probabilities of meeting all these conditions, they multiply. And you will become horribly sample inefficient. So what we want to do is to already guide the generation process towards something that only binds to A and not to B, C, D, our off targets. And yeah, there we're working on some algorithms right now where you can use some diffusion model steering methods to actually sample this distribution of binding to A and not to B, C, D. And if we want to go into that a little more, I'm happy to say more about that. But otherwise, I'll let you moderate and decide.
Is that already in GitHub?
That's not in the GitHub yet. That's
okay.
Something we still work.
Yeah, let's see how many questions do we have. We only have one more question. Yeah, I think we can go a little bit into that if you have time.
Yeah,
we don't want to miss your. Yeah, I think this is the same person who asked the question. Yeah, do you want to ask a qualifying question? Go ahead.
Hi, thank you so much for the great presentations. And I'd like to follow on that because I see in the input YAMLs, you already have the features of specifying we can give it additional chains and ask it to design the binders that not bind to that chains at all. So do you think that can be a proxy or a way to do anti-screenings against that chain?
Yeah.
Or how does it work in actuality?
Okay. Then, so Wei Yang's question, I will just repeat it a little bit. We use now this not binding feature to control our selectivity, our specificity also to off targets? And yes, I think in certain scenarios, in special cases, we can use this, right? If we, for example, have this glycan protein case here, where we only want to bind the version that has the glycan, we do not want to bind the version that does not have the glycan, then we can guide the model to generate something that only has spatial proximity to the glycan and doesn't have spatial proximity to the rest of the protein. Because otherwise you might be making interactions with the rest of the protein and then also binding to the protein by itself, even if it does not have the glycan attached to it. Now, this is something where we're not explicitly then guiding away from not binding the protein, but we're just we're not saying generate something that does not bind, but we're saying generate something that binds the glycan. And then you would hope that if you only steer and guide toward binding the glycan, then you don't end up with something that also ends up binding the protein. Let me know if that didn't clarify, then happy to go into that a bit more and talk about the other scenario that I'm using here.
I think this is fine, but maybe talk about the other scenario as well. Because this is very interesting for orthogonality and getting heterodimers that are orthogonal and stuff like that.
Yeah. Oh no, we all love our selectivity. Actually, where's now my presentation gone? Here we are. And yeah, it would be nice to have this. We all know what selectivity means here probably. And now what I'm working on with my student, Nuran Wang, who's pushing this project, is to have an algorithm where I will just remind us here, right? We have this diffusion model where we iteratively step toward generating something, or we iteratively update our 3D coordinates to then end up with this final 3D structure that defines our binder. And the direction that we step into is given by this gradient of the log density that our model tries to approximate, our diffusion model. And now we can say the gradients, the direction of steepest increase of the probability of binding to A. And A is now this target here, and we want to increase it, right? So we step into this direction of steepest decrease. And we can also write down this direction for another target. Maybe if this is our off target, then we would sample from this region here and not from the middle region where you would have likelihood for interacting with either target. And now, what's one naive way to solve this would be to use classify-free guidance, this technique from diffusion modeling where you essentially just step away from binding to B and toward binding A. But this wouldn't work for several reasons, like that if you're close to already have high likelihood for binding to B, then you will not be actually stepping away because you're close to a maximum and then your gradient will be very small. Now just being conscious of the time here and how I should handle this. And we'll say that what we would like to sample instead is something like this here, where we say we want to bind to, or we always want to meet a threshold for being more likely to bind to A than being likely to bind to B. And this is actually something that we can sample with techniques that combine sequential Monte Carlo with diffusion models. And yeah, that's something we're still working on. But with that, I would maybe leave it to follow-up questions on it or to move on to our question if you recommend pursuing such.
Very cool. I think we can probably go on in the interest of time. We have 7 minutes. And maybe one question from Jennifer, maybe what's your favorite metric? I think this is very like a subjective question, a favorite metric. Did you find any metrics that are very predictive of binding? Is maybe a better general question.
Like, not right. We always mush up all of our metrics into this final ranking, and we then of course do some attributions afterward to see which metrics are the best at ranking individually. But the thing that comes out there, if you use a single metric, then you want to be the most powerful, still IPTM, like min interaction, PAE, IPTM, and so on.
Okay, then Atarva, do you want to ask your question in person? Then just unmute and fire away.
Hi, Anas. I have a quick question. Actually, when we design a binder, can we somehow account for the things like environmental condition like temperature, pH, or buffer condition? Because sometimes the design look good computationally, but don't work well in the wet lab condition. So is there any way to control or set the parameters like temperature or pH so that the design binder will work based on a specific condition or will fold and be activated at specific conditions?
No. So it's right. We are at the stage right now where we're trying to design something that binds as strongly as possible at averaged over all the pHs that you see in PDB and as stable as possible. And we're then we're not trying to control somehow the temperature at which it binds, but we're just trying to make the thing as stable as possible, as tightly binding as possible. And hopefully all pHs, but probably pH dependent, and the most for, I assume, physiological pH of 7.4 is what you observe the most in RCSB. So that's what the model has seen the most. But this is an incredibly interesting question. And I think this is something where machine learning soon could really have a big impact, or which feels particular to, for example, this pH-dependent binding question, right? Where we could now imagine wanting to do something like this, where we say we want to have a binder that's at physiological pH of 7.4 does not interact with the target, but pH 6, which may be the pH in the tumor microenvironment, there we would want then to bind, and then we have a design that only interacts with the target and has this mechanistic effect on the target only in this tumor microenvironment. Or this is also very interesting for endosomal escape or for getting through the blood-brain barrier, this pH-controlled binding question. And I think there is now actually a lot that we can do for reasoning about this biochemistry problem. And what are the residues that change their properties as the pH changes, like changing their charge? And then how can we place them correctly with respect to charges, for example, in the target, such that we make or break interactions in the threshold that we want? And all the computational work that I've seen done in that space so far is randomly placing more histidines in our inverse folding model, like increasing the histidine bias in our inverse folding model. And then looking at, does it happen to make interactions with the target? And there's so much more that we could do there with guiding the geometry, and also not just histidines can be in this right range of pH dependence. We could bring design the electrostatic environment around another residue such that it has this property of changing its charge, for example, in the right pH range. Okay, but I think that's a very nice question.
Very cool. There's also HBNet from a while ago, where you can design hydrogen bond networks with histidines and then hope that when those break, they go away. So maybe a question, if you don't have enough GPUs or if you're compute limited, is there a parameter that you can retune, have a trade-off between accuracy and the time it takes for compute, reduce the number of steps in the denoising process or something like that?
So unfortunately, the denoising process is not what takes the most time. Yeah, I want to caution against it, right? Because we also had, for example, this Nipah virus design competition, and then a lot of people submitted their design or 10 designs when generating 10 design with the model, and then you just will not get good designs. And it's unfortunately the case with this model that you have this large amount of samples. Now, you've seen something like Proteina Complexa from NVIDIA, for example, coming out, right? Where they try to steer the model at generation time already towards satisfying these refolding metrics and such to be more efficient. Now, the question is, how much more efficient is it actually than in practice? It turns out that it's not going far beyond best of n sampling. And one issue why this is the case is because these inference time steering methods that they use for diffusion models, they always operate on this, on what's called the denoiser in our diffusion models, where you look at the average of what you would get out at the end, and then put this into your reward function that then ranks it and tells you how good it is. But this average is then an average between two protein structures or two protein sequences, which might be completely nonsensical. So you don't get very much steering, very much guidance. So there, if you're interested in something like this, I would recommend looking into methods like diamond maps, which is a paper in the flow map guidance and steering literature that came out recently. Diamond methods.
And do you have a rule of thumb of how many designs it's good to run? I guess the answer is as much as possible, but
no, I mean, you can do things like the ideal thing would be if you make yourself a training curve, right? If you look at what are your metrics that you're getting and when do they saturate, and then you have how many samples you generate on the x-axis, and then your scores on the y-axis, and then you see I don't need to generate any more samples because the metrics are saturated anyways. And something like this, like getting a curve like this, I have not implemented in the BoltzGen repo, but that would be certainly a nice user help. Also, I think for RF diffusion, for protein complexa, for any type of model like this, if someone builds something like this.
Yeah, very cool. That's a good idea. Can this framework handle intrinsically disordered proteins? I guess, yeah. You showed some examples at the beginning, right?
Yeah, we have experimental validation as well, where we, for example, have this intrinsically disordered fusion oncogene and then design binders against it and find that one of the 9 designs is successful. Or we also have some SPR BLI for intrinsically disordered protein binding and get 269 nanomolar and 296 nanomolars for when testing 10 design. Yeah, stuff like that.
Ooh, very impressive. And
the numbers that I just mentioned aren't actually in the paper yet, but we'll update that soon. Sorry.
Maybe like a final question. Is the refolding model of BoltzGen basically the same as Boltz2 standalone?
Yep, it's exactly the same. Except it doesn't, as you already said in the previous talk, it doesn't use multiple sequence alignments.
Cool, because there's no... Oh, do you need multiple sequence alignments for the target, or is that usually not a problem?
No, we don't use multiple sequence alignments for the target, and we either run Boltz2 to get the structure and then feed in the structure of the target to BoltzGen. And right, if you already have the structure for the target, you don't need the multiple sequence alignments.
Okay. Oh, because it's not regenerating it, or is it regenerating it at the design
It is
time?
regenerating. It is regenerating the structure. But if you have the structure as a covariate input, this also tells you this contact information that you would get from your MSA.
Oh, you're using it like a template, right?
Correct. Very similar to a template.
Very cool. Very cool. Great. Thank you again. Thanks again to both speakers. These were amazing talks. And so, yeah, would you be willing? Are you fine with us putting the recording on YouTube?
Yep, absolutely.
And will you be willing to share the slides like in PDF form, and we'll put them in the description of the video?
Sounds good.
Okay, perfect. Perfect. Great.
Then, Dasta, for me, big thanks for making this happen and holding these seminars. And to everyone else, thanks for the amazing questions.
Thank you, everybody.